Okay, let's continue on with our discussion of how gas is exchanged in leaves. Write in your notes that the exchange of oxygen and carbon dioxide happens in the leaf, as well as the loss of water vapor in a process called transpiration. Now that's done through pores we call stomata, the singular of which is stoma. We also said that normally stoma open when the light strikes the leaf in the morning, and then they close during the night. So make sure you have that in your notes as well. Now we also want you to get in your notes that the pores are opened and closed by structures called guard cells. And you see those guard cells now right here. So here is the stoma, all right? And here are the guard cells that open and close it. So make sure you get those in your notes. Might be a good idea to actually sketch this in your notes so you have it. So go ahead and make a rough sketch of the guard cells. And notice that the chloroplasts are contained in the guard cells as well. So when it's open, this is referred to as an open stomate, or one stoma, or a closed stomate. So let's get the terminology correct here before we move on. So these stoma are the source of the carbon dioxide that the plants will use in the Calvin cycle. Make sure you have that down. It's worth looking at the chemical equation for photosynthesis and making some sense out of it now that you know what you know about the light-dependent reactions. So let's take a look at it. We have it right here, conveniently, huh? Okay. Now, so the reactants are water and carbon dioxide. And if we look at our arrow over here from water, water is extremely important. So we see now that water is split in the light reactions by what? By the water splitting enzyme. Now, hydrogen now, notice that they have hydrogen blocked off here, is combined during the dark reactions to form pGal. We're not going to write that down yet. Excuse me, but that becomes really important, this pGal molecule. You'll understand here in a bit. We know that oxygen is given off or released into the air. We also know that we use those electrons in water to replace the electrons that were lost in photosystem 1. So we know how important it is. Now here's our carbon dioxide. Carbon dioxide now is taken in from the atmosphere through these stomates. Okay, so there's the point there. And then we know that light is from the sun. And we know that it's used to split water to form ATP and NADPH. Now over here on the right-hand side now, here's our organic molecule, C6H12O6, that's glucose. And here again, they mentioned pGals, and we'll see what those are here in a bit. But it's this carbon dioxide molecule that will help form and reform this glucose molecule. So, question for you. Entering the Calvin cycle are now 3 important substances. Can you name them? If you said ATP and NADPH from the light reactions and carbon dioxide taken in through the leaves, you'd be correct. Now let's do an overview of the Calvin cycle and see if we can start to unravel a fairly involved process. Remember, we're now in the stroma of the chloroplast, not in the thylakoid. So as we see here, we're in the stroma, all right, not in these disc-like things called the thylakoids. That's where the light reactions occur, right? So we're here in the stroma, not in the thylakoids. Now, let's write some general steps in this process first, and then see them work using this simulation. So, here we are in the stroma that surrounds the thylakoids, not in the thylakoid membranes. It's also worth mentioning that this is the Calvin cycle, meaning that it will end up where we start. That's the idea behind a cycle. So, we're in the stroma of the chloroplast. Now, step 1. Carbon dioxide is added to a 5-carbon molecule called RUBP. There it is right there. So carbon dioxide, here's our CO2, is added to a 5-carbon molecule called RUBP. RUBP is the long name. You don't really care about that much. They're going to use RUBP in the exam. So, now this results in a 6-carbon molecule. That makes sense. There's 5 carbons here, and there's 1 here in this carbon dioxide molecule. And this results now in a 6-carbon molecule, which makes sense. However, this 6-carbon molecule is extremely unstable, which means it only lasts for a brief second. All right, so again, carbon dioxide combined with RUBP to form this temporary 6-carbon molecule. Now, step 2. The 6-carbon molecule now splits into 2 3-carbon molecules. And we're going to give them a name here. They're called 3-phosphoglycerate. There you see it. So 2 3-phosphoglycerate molecules. So the 6-carbon compound splits into 2 3-phosphoglycerate molecules in step 2. Now, step 3. Now we'll use the products from the light-dependent reactions. Remember, there are 2 of them, NADPH and ATP. In step 3, we use the energy in ATP and the reducing power in NADPH. So the 3-phosphoglycerate molecules move through a series of reactions and are converted into 2 molecules of glycerol 3. There's our ATP and NADPH. To 2 molecules of glyceraldehyde 3-phosphate. So, that's kind of complicated name-wise. So the 2 3-phosphoglycerates combine with ATP and NADPH to form 2 glyceraldehyde 3-phosphates. Now, that's also G3P. They're called G3Ps. Glyceraldehyde 3-phosphates are also called G3Ps. So get that in your notes as well. Now, step 4. When several of these G3Ps or glyceraldehyde 3-phosphate molecules have been produced, some combine to make glucose, while others are reused in the Calvin cycle to form the original RUBP. So here's our G3Ps. Here's some of them forming glucose. They'll be used to form glucose. And some now, here we go, the other ones, are used to regenerate RUBP. So here's our RUBP molecule. So write that down in step 4. Now, that's a very general look at the Calvin cycle. Very, very, very general. But it kind of gives you an idea of the inner workings of this process. So again, here's our carbon dioxide and our RUBP combining to form that temporary 6-carbon molecule, which breaks into 2 3-carbon phosphoglycerates. Those then are combined with ATP and NADPH to form 2 G3Ps or glyceraldehyde 3-phosphates. Some of those go to form glucose while others reform the RUBP molecule. So there's a general idea of what's going on now in the Calvin cycle. Okay, now here's another diagram of the Calvin cycle with a bit more detail in it that we want you to get in your notes. And you're going to find out that when we're done with this diagram, there's going to be a tremendous amount of detail in it that's not there now, but we'll kind of give it in small doses. So let's go back to your steps that you just wrote down and add to them where necessary. So let's look at step 1 first. Recall that a 5-carbon molecule called ribulose biphosphate, or RUBP, binds carbon dioxide dissolved in the stroma. All right? Now, so here's RUBP combining now with carbon dioxide. All right, now write in your notes that this is called carbon fixation, or fixing the carbon molecule. Now, and it's catalyzed by this enzyme, rubisco, ribulose biphosphate carboxylase. All right, so write that in your notes as well. This is this you'll see this on the exam for sure. Rubisco is called the miracle molecule, and we'll talk about it here in a second. So, this 5-carbon sugar, RUBP, fixes the carbon in carbon dioxide, and rubisco now is the enzyme that facilitates it. Remember what enzymes do? They reduce the amount of activation energy required for this fixing process. So write that this enzyme rubisco facilitates this carbon fixation. So now, 5 carbons plus 1 carbon equals this temporary 6-carbon molecule. Now, also recall that this molecule breaks down to give 2 molecules of the 3-carbon 3-phosphoglycerate, or 3PG. Okay? So, let's take a look at that. So here's our 3 carbon molecules now, called 3-phosphoglycerate, but they're also referred to as PGA. Phosphoglycerate is also referred to as PGA. Now, so 3 names for this, right? Leave it to science, right? So it's called 3-phosphoglycerate. It's also called 3PG, and it's also called PGA. So make sure you get that in your notes as well. Okay. Now, you need to know both names in case they are asked for on the AP exam. In fact, all 3. Most times you're going to see the thing called PGA, but you may see it called the other 2 names as well. So make sure you have those in your notes. All right, now step 2. Recall that these 2 PGA molecules are converted into glyceraldehyde 3-phosphate, right? Into glyceraldehyde 3-phosphate. Add to your notes that this molecule is also referred to as G3P, not 3PG, it's G3P, glyceraldehyde 3-phosphate. So there it is. Now, it's also referred to as PGAL, PGAL. You may see that on the test as well, PGAL. So write that in your notes. So this G3P is also referred to as PGAL, PGAL. So write that in your notes. PGAL is a 3-carbon sugar phosphate formed by adding a high-energy phosphate group from ATP. Okay? So here's our ATPs down here. Then breaking the phosphate bond and adding a hydrogen from NADPH. Here we are, NADPH. And NADH is now ready for another hydrogen to be added to be used over again. Remember, we got that NADPH from the Calvin cycle. So it's going to go back, or from the light reaction, it's going to go back to the light reaction to get phosphorylated again and have this H added onto it. This ADP will also go back to the light reactions and be phosphorylated to form ATP. Now, here's our G3P molecules. Let's go to step 3. Write in your notes that 3 turns of the Calvin cycle, which means you're using 3 molecules of carbon dioxide, 3 turns of this Calvin cycle, right, produces 6 molecules of G3P, 6 molecules of PGAL, or G3P. Also write that only one of the 6 molecules exits the cycle as a glucose molecule. So only one of the 6 G3Ps or PGALs becomes a glucose molecule. Now, so the The other 5 enter a complex process that regenerates more RUBP. We saw that in our basic diagram. So 1 glucose is formed and 5 G3Ps go back to generate now more RUBP in this process. Now, you can also see that ADP is now available to be converted to ATP again and used in the next turn of the carbon cycle. So, important for you to understand the cycling process. It's complicated, but not that bad. Now, we're back to the beginning of this process with the formation of RUBP again, right here. Okay. We also now have ADP available to convert back to ATP, and NADP now ready to convert that back to NADPH to start this reaction over again. Whew! Okay. Take a picture of your notes, submit them to Moodle, grab that Part 4 study guide and answer the questions, and I'll see you back here for Part 5. Bye.